Okay, I just wanted to start by introducing myself, and I believe, Sammy, this is is this through the REC program, through the research education component?
Yes, so there's many rec scholars on here, but it's also invited to all faculty across BU-ADRC. So you have a mix of both.
Perfect. So I did just want to tell you a little bit about my career trajectory. I also lead the research education component at our Wisconsin Alzheimer's Disease Research Center. So I think it's always interesting to hear a little bit about how people got where they are. So I completed my undergrad in psychology at the University of Winnipeg in Canada. And then I moved to Arizona and had a couple of years where I worked as a research assistant in a lab before I was accepted to the University of Arizona graduate program, where I worked on neuroimaging studies related to aging and Alzheimer's disease. Then I came to the University of Wisconsin in 2007, where I completed my postdoc. And then I was promoted to a scientist, and then I started my faculty position in 2010. So I just want to share that one of the things I really love about doing research at the University of Wisconsin, but more broadly in Alzheimer's disease, is that I think our field is really collaborative. And so I've had the pleasure of being able to collaborate with a number of great investigators who are doing things that are very different from me. And I'm going to tell you about some of that today in some of our work on gut microbiome because it's very transdisciplinary. So let me get started.
Yeah, and thank you so much for that.
Absolutely. Just gonna full screen this. Hang on a sec. All right. So I'll give you just a little bit of introduction into how we got into this work, microbes and neurodegenerative disease, some recent findings, some of the interventions that we could potentially test if gut microbiome is a modifiable factor, and also future directions. So this is Elia Mechnikov. And Elia Mechnikov won the Nobel Prize, I think in 1908, in the early 1900s, for his work on discovering macrophages. And another thing that he is perhaps not quite as well known for, but is a little related to his work on the immune system, is that he was really interested in aging and longevity. So he actually coined the term gerontology. And part of his interest came from observing individuals in Eastern Europe who were very long lived. And he was interested in what people were eating that might contribute to their longevity. And he saw that a lot of these centenarians were eating cultured yogurt, that they were eating sour milk and yogurt. And he actually developed a theory that if you could ingest a lot of microbiota, that it might crowd out microbes that cause poor health. So he himself started to consume sour milk and yogurt. And at the time that he was interested in this topic, there was actually a cholera epidemic, and he had this theory that some people were not getting cholera because they were basically crowding out bad bacteria with good bacteria. So, as part of his own experimentation, he infected himself with cholera, and he did not get sick. And he was really a proponent of this idea that there can be probiotics. So he's actually kind of the father of gerontology and the father of probiotics. So I thought that was very interesting and a nice intersection to what I wanted to talk about today. Because, of course, it's very interesting to see different trajectories of aging to determine why some people can live to be a very old age and have good cognitive health and not develop neurodegenerative disease. And then why do other individuals develop dementia and diseases of aging? So one of the concepts that kind of I think is related to this is the idea of the exposome. We of course know that genetics are incredibly important in terms of determining health outcomes. They play a role in longevity, they play a role in neurodegenerative disease, but we know that genetics doesn't explain everything. So, for example, there's cases of genetically identical twins where one might develop dementia and the other person might not develop dementia for many, many years later. So there might be some other factors that are predicting onset of dementia. The term that kind of encompasses this is the exposome. So it complements the genome. And the exposome is all of those factors that people are exposed to from in utero all across the lifespan until death. And some of the things that you could think about would be, let's see, can I do a pointer here? So some of the things, exposures that people think about are living environments. We do a lot of work in Wisconsin on area deprivation index and the neighborhoods that people live in and how those contribute to health outcomes. You could think about the food that people eat. But of course, this also is not just what we call a lifestyle factor or a behavioral factor, but could also be influenced by what kind of neighborhood do you live in that might have access to healthy food? Are you exposed to smoking or pollution or pollutants in the environment? Do you drink alcohol? That can also be influenced by the types of environments that you're living in and also quality of sleep. So we study a lot of these factors in Wisconsin, and another exposure is actually the microbiota that live in and on our body. They are part of us, but they are also in some ways not a part of us, of course, because these are non-human cells. And the microbiota, there's about one microbiota for every human cell in the body is the approximate estimation of how many microbiota we have. And a lot of those live in the gut. And we actually think that maybe even some of these exposures that we have across the lifetime could act via the microbiota because we know that the microbiota are very important for health. They're also involved in disease, but they're also involved in supporting our health. And so just as one example, for example, individuals who work in environments where they're exposed to a lot of pesticides, they actually have a depletion of microbiota in the gut microbiome, and that could potentially contribute to metabolic disorders. So this is one of my favorite stories about the gut microbiome and kind of illustrates a way that we can study the gut microbiome and understand how it's related to health and disease. This project was conducted by my very good friend and collaborator, Federico Rey, who I met just over 10 years ago at the University of Wisconsin. And he was involved in a really interesting study that was trying to determine how the gut microbiome is related to obesity. So he studies germ-free mice. These are mice that have never been exposed to any microbes at all. So they're kept in these containers where the food is autoclaved and the water is microbe-free. And the kind of nice thing about these models is that you can then introduce microbes and see how they influence health outcomes and how they influence the phenotype. So in this study, the investigators were interested in how obesity can contribute to the phenotype, the microbes that we see that might be related to obesity. So first of all, I'll tell you that if you take a germ-free mouse and you feed it a high-fat, high-sugar diet, the mice don't gain weight and they don't develop metabolic disorders. But then if you humanize the mice by collecting stool samples and giving them to the mice, then you can see if these these communities that live in humans, if those contribute to the phenotypes. So what they did was they collected stool samples from twins who were discordant for obesity, where one twin was deemed obese and the other twin was so-called normal weight. And if you take stool samples and humanize the mice that are humanized using stool samples from the so-called normal weight twin, if you then feed them a high-fat, high-sugar diet, the mice do not gain weight. They do not develop metabolic disorders. But if you humanize them using stool samples collected from someone who is obese, then the mice start to accumulate adipose tissue and develop metabolic disease when they're fed a high fat, high sugar diet. So there's really something about the gut microbiome composition that seems to be affecting the phenotypes in these mice. So when Federico and I met, my lab was actually kind of interested in type 2 diabetes and how it might be related to brain health. And we thought, well, perhaps some of the effects of some of the risk factors for dementia could potentially be acting through the gut. So, how do we study the microbiome in relation to some of these outcomes? Well, I did tell you about this germ-free animal model. Um, we are in our research program, we're collecting stool samples, and you can do 16S rRNA-seq to find out which bacteria are present. If you do metagenomics, then you get a more comprehensive evaluation of what's present in the gut, also beyond bacteria, including viruses and fungi. And you also get function of the microbiota that are present. There's all kinds of other analyses that we can do on the stool samples, including proteomics, lipidomics, foodomics. There's researchers who are using stool samples and can then determine what people are eating based on the metabolites that they find in the stool. And then you can do, as I mentioned, you can do these germ-free mouse models, or some people are also using antibiotics to wipe out the microbiome in the mice for experiments. So in our first study, our idea was to perform mechanistic experiments in germ-free Alzheimer's mice. So we started some colonies of mice that were bred to overproduce amyloid, and we kept them germ-free. And the idea was to see what happens if we keep an Alzheimer's mouse germ-free. And then we also had a human component to see if gut microbiome differed between people with and without Alzheimer's disease. So to start off with kind of the first question, right when we were planning these experiments, some papers started coming out already. So in this paper from Sam Sisodia's group, when they used antibiotics to wipe out the microbiome in these mice, they found that those mice that had a wiped out microbiome, that they actually had less amyloid plaque in the brain. And likewise, then in some studies that looked at germ-free mice, which was our intention as well, when you look at germ-free mice, they also show less amyloid plaques in the brain. So this just sort of like, these are very kind of high level experiments. They don't tell us a lot about mechanisms, but they do tell us that there's some link between the gut and the brain pathology. In our human experiments, we leveraged some ongoing cohort studies. So we have a Wisconsin Alzheimer's Disease Research Center. So we have a clinical core, and those research participants undergo a number of measurements, brain imaging, including MRI and PET, comprehensive cognitive testing. Many of our participants undergo lumbar puncture so that we can look at the proteins related to Alzheimer's disease in the cerebrospinal fluid. And of course, as all of you know, there's also developments now in the blood biomarker space. So many of the proteins that previously we only measured in the cerebrospinal fluid, you can now measure in blood as well. These are some of the proteins that we look at in the cerebrospinal fluid and in blood as well. Some of those include measures related to amyloid and tau pathology, including phosphorylated tau protein. We look at neurofilament light chain, which is a marker of neurodegeneration. In CSF, A beta 42 over 40 as a marker of amyloid. But you can see there's a number of things that you can measure in the fluids that serve as really good outcomes of what we're seeing in the brain. So in this very first study, we recruited people with dementia, mild dementia due to Alzheimer's disease, as well as people who were cognitively unimpaired. And then we had a number of exclusions related to gut microbiome. And one of those is antibiotic use within the last 6 months. And actually, in our recruitment efforts, we found that a lot of older adults are taking antibiotics. It might be for health conditions, but sometimes it's because they have a hip replacement, and they've been told that they should take antibiotics before a dental cleaning. This has been very, very common, even though the guidance on taking antibiotics before dental cleanings has changed. We still encounter that quite a lot in our program. So for this first study, we performed 16S rRNA-seq, and we looked at diversity of the gut microbiome as well as abundance of gut microbiota. And if you're if you sort of think about what would make up a healthy environment, typically we think that diversity is a good thing. So if you're thinking about a healthy ecosystem, you want lots of different plants and animals versus, a depletion of diversity would mean that there's only like a few kinds of microbiota there. The most extreme example of that would be something like C. difficile, which is a hospital-acquired infection where C. diff really takes over the gut and then can cause pretty significant health problems. So we found that in Alzheimer's disease, there was reduced diversity. There was a number of taxa that were, there was a few, I should say rather, there was a few taxa that were increased in Alzheimer's disease, but the majority that we found that were different were actually decreased. So that sort of makes sense too with this picture of reduced diversity. That's also been replicated in a number of studies that came after ours that in Alzheimer's disease, you see reduced diversity. That is perhaps not super unexpected. In many diseases that are studied in relation to the gut microbiome, reduced diversity is kind of a common outcome, and it's not really specific to Alzheimer's disease. In Alzheimer's disease dementia, you also see that people are starting to eat differently and that they're also taking medications to treat Alzheimer's disease, and those can also have an effect on the gut. So one of the things that we did was to also see whether the abundance of certain microbiota was associated with brain outcome measures that we looked at in CSF, even among people without dementia, because those would be individuals who, for example, maybe haven't had diet changes, they're not taking medications related to Alzheimer's disease, and we wanted to see if there was an early relationship. And in fact, we do see that there are relationships between abundance of certain microbiota and the level of certain proteins that we measure in CSF, even among people who are cognitively impaired. When we look at everybody together, so all of the participants, both with and without Alzheimer's disease, the relationships are stronger. But we do still see some relationships, for example, with amyloid and phosphorylated tau measures, even in the asymptomatic individuals. So how does the gut impact the brain? Well, since I started this work, I've learned a lot about the gut microbiome because it's not something that I trained in. But this is kind of outlining some of the common mechanisms that people are looking at that link the gut to the brain. So first of all, we know there's bidirectional communication from the gut to the brain, but also the brain to the gut. Much of that is via the vagus nerve. There are many microbial metabolites, so small molecules that are produced by the gut microbiota or are modulated by the gut microbiota that then go into systemic circulation and signal with the brain. One of the mechanisms that we've been really interested in is gut permeability. And when I started this research, Federico had suggested that we look at leaky gut as a mechanism in neurodegenerative disease. But at the time, I felt that was much too fringy. But since that time, we've actually generated some results that suggest that in Alzheimer's disease, that there might be gut intestinal permeability. One idea is that there might be translocation of bacteria or bacterial products from the gut that then get into systemic circulation and maybe even the brain. I would say that in general, there seems to be very little evidence that microbiota are actually getting into the brain, but bacterial products is quite possible. So, for example, LPS, lipopolysaccharide, is a component of gram-negative bacteria, and we know that that gets into systemic circulation. The microbiota also play a very strong role in the development of the immune system, and they continue to signal the immune system over your life course. So we do think that the microbiota are probably in the case of Alzheimer's disease, that there is some immune function involved as well. With regard to some of the gut permeability studies, we had an R01 grant funded to look specifically at intestinal inflammation. So the idea being that with reduced diversity, you might have altered, you sort of get a intestinal epithelia that's not healthy, and you get intestinal permeability, and that could then in turn exacerbate Alzheimer's disease pathology. And this is my colleague Tyler Euland, who's done some studies to understand how immune signaling then can lead to increased spreading of amyloid pathology in the brain. So he was one of the investigators that discovered disease-associated microglia and how they are involved in spreading amyloid through the brain. So this is one hypothesis that we've been pursuing. So we use the data that have been collected from research participants in Wisconsin, and we add the stool sample collection as well. When the stool samples are collected, they're then shipped to Federico's lab, and they come in these tubs. It used to be the case that we would just get the tubs and put them in the freezer. That, by the way, is not very practical for when you want to do further analysis of the stool samples, because you have to take, I'm not kidding you, a chisel and a hammer to knock off pieces of the stool. So it is a lot of work. Federico's lab developed a better technique called the FAST technique, which they essentially just use these sterile straws and they dip them into the stool and they fill up the straw, and then they aliquot the straws at -80. And now it's much easier to just cut off a piece of the straw and do analysis. And again, using the stool samples, we store quite a lot of stool from all of our participants. Some people choose to just get like a small sample, and then you can do metagenomics on that sample. You can also do other analyses, but of course, always the more sample you have, the more things you can do into the future. So one of the things that we looked at in these stool samples is something called fecal calprotectin. It's a protein found in neutrophils. And when measured in stool, it's an indicator of intestinal inflammation. And it's elevated in inflammatory bowel disease. We didn't really have participants in our research study who had very, very high levels of calprotectin, but we did see a range of calprotectin levels in our study, and we checked to see, first of all, is inflammation higher in people with Alzheimer's dementia? Is it higher in people who have amyloid in the brain, but no dementia? So this idea of like preclinical Alzheimer's disease. And then is it also associated with cognition and Alzheimer's disease outcomes? So this is just to kind of illustrate the different points we were looking at. So we're looking at individuals who have no amyloid. They might be over here. Then we have people who are preclinical, who have some amyloid in the brain, but no symptoms. And then we have people who have dementia. So if we just first start by looking at age, you can see there's a lot of variability in calprotectin, but as people get older, we see that there's more calprotectin. And that's true whether we include people with dementia in our sample or we just look at the people without dementia. When we look across these different groups, we see that only people with dementia have elevated inflammation compared to our cognitively healthy participants. So we didn't actually find that in people who are cognitively healthy and have amyloid in the brain, that those individuals have intestinal inflammation. It was really only in the people who already have dementia. And then when we looked at amyloid PET imaging, what we found was that in that Alzheimer's group, as people had higher levels of calprotectin, that was also associated with more amyloid in the brain. And one idea could be that in some way intestinal inflammation is exacerbating amyloid or amyloid spread. The other possibility is just that people who have more amyloid in the brain have worse disease, and so for some reason that is then affecting the gut and causing inflammation. We don't know from our human studies what the relationship is, but Tyler's lab is now doing the animal studies to see what the mechanisms are. So inducing inflammation in Alzheimer's mice, and seeing what the relationships are. We also looked at the CSF biomarkers. So we do see that higher inflammation is associated with more amyloid measured on the biomarkers. And then with regard to neurofilament light chain protein, there's a modest relationship. The p-value is behind the screen, so I do not believe it was significant though. Oh, it was. It did reach significance. So we do see a relationship with neurofilament light chain protein, which is a marker of neurodegeneration as well. When we looked at the cognitive function, again, we find that with higher inflammation, there was worse long delay recall. So on the Ray auditory verbal learning test, this result was significant, but not with learning or with a processing speed test. We have started to also see if we can analyze whether the stool itself has measures of inflammation. So there is a platform that's gaining a lot of popularity for measuring blood biomarkers of Alzheimer's disease, which is called Neulisa. It's very, very sensitive. So we have looked at they have an inflammatory panel. And so we've run some of our stool samples on the inflammatory panel. Just in some preliminary data, we looked at healthy controls versus amyloid positive individuals with dementia. We have found that people with Alzheimer's disease do seem to show differences in the inflammatory panel. Also, we were just very pleased to see that we were able to pick up and get a lot of hits in the inflammation panel in stool, because if stool hasn't been, I think we might have been the first to run it on the Neulisa panel. So the Neulisa panel is actually optimized for blood and CSF and not for stool. So just the fact that it worked was quite satisfying. And then some of the differences were found in inflammatory markers that were related to actually blood-brain barrier, neurogenesis, and inflammation. We've also been interested in whether ApoE4 differences are observed in the gut microbiome. It is the case that actually, based on genotype, that we see differences in human gut microbiome, even among people who are cognitively unimpaired, which has raised an interesting question for us, which is whether some of the effects of the ApoE4 genotype could be mediated by the gut microbiome. So in a recent publication, we found, a recent study rather, that was published last year, we found that ApoE4 carriers showed a number of differences in the gut microbiome, including Firmicutes-related differences, also differences that could suggest that people with ApoE4 might have more pro-inflammatory metabolites produced by bacteria and lower amounts of short-chain fatty acids. So we are now following up on those results to see if we can more mechanistically understand what these differences in ApoE4 carriers might mean. We've also looked at short-chain fatty acids, which are produced when we eat fiber-rich food. And this might be one of the mechanisms by which fiber is good for us. So much of the fiber, it's not necessarily just for us. It is for feeding those microbiota. And when you have higher levels of short-chain fatty acids, then you have better gut barrier integrity. There might also be some anti-inflammatory effects. And what we've seen in our studies is that when there are higher levels of short-chain fatty acids in the stool, that is actually associated with lower amyloid levels in the brain and better cognition, which again seems to suggest that there might be some ways that we could potentially modify the gut to improve health outcomes. So far I've talked about the microbiome being altered in Alzheimer's disease, some relationships with gut inflammation, genotype effects, and then the fact that these molecules are very important, the ones that are both produced by the gut or potentially modified by the gut. So now that we know that the gut is different in Alzheimer's disease and aging, how do we modify the gut? So I have a list here of many things that influence the gut. These include genetics, early life experiences, how old you are, where you live, who you live with, what you're eating, are you drinking alcohol? With regard to who you live with, my colleague Federico did an interesting study where he found that they were studying people who had lived together as siblings and then looked at what their gut microbiome looked like in later life, of course, when they weren't living together anymore. And what they found was that people tend to look like the people that they're living with, maybe because there's microbial exchange or because you're eating the same food. But spouses tend to look more similar than, say, siblings who lived together when they were younger. And spouses who like each other tend to have more similar gut microbiomes. Of course, if you take antibiotics, that influences the gut. It reduces diversity, but much of it bounces back over time. Probiotics can influence the gut, gastric bypass, disease. Pets is a fun one. So if you have, for example, dogs that are coming in and out of the house, they'll change the types of microbiota that are in the house. One of my favorite studies about gut microbiome change has to do with immigration. So this was a really nice study done by Dr. Van Gay when she was at the University of Minnesota. And she was interested in immigrants coming to the United States. And she worked with Karen and Hmong individuals who she studied in their native Thailand, but then also studied new arrivals to the United States, as well as long-term residents and the second generation of kids. And what she found was that Hmong and Karen people living in Thailand had much higher gut microbiome diversity and higher prevalence of fiber degrading enzymes. But then when individuals moved to the US, then they lost diversity. They also started to get more evidence of metabolic diseases like obesity and type 2 diabetes. And that's exacerbated by the longer time that you live in the US, and you see the greatest prevalence of these metabolic disorders among people who are second generation, who also had the least diversity. So she did a really nice project because it was really a community-based participatory research approach where she interviewed her participants and she was really interested in kind of some of their insight into the science. And she thought perhaps it was the case that when people moved to the US, they just stopped eating certain kinds of foods. And there was that to a little bit of a degree, but actually, they were still seeking out many of the same foods when they moved to the US. So she determined that it wasn't really explained completely by diet. So there could be other reasons why, when people move to the United States, that there's a loss of diversity, whether it's water, pesticide exposure, or stress, or really any number of things. But it was really because she had been talking closely with her participants that she was able to get more insight. And she also did a really nice job of returning results to her participants and also starting some cooking classes and other things that really made it a wonderful project. How do we modify the gut microbiome? So some of the approaches that people are taking include fecal microbiota transplantation. We actually tested the first FMT study in Alzheimer's disease here in Wisconsin. It was during the pandemic, but we used a intervention that was an oral microbiota transplant. So these were actually stool-filled capsules. And for a full transplant, it was 30 capsules. And participants were actually very interested. And the idea is not that we would, say, treat Alzheimer's disease with FMT, but what we were thinking was that if you modify the gut, then you can see if there's any relationships with the brain. What were the modifiers that led to brain changes? And perhaps those could be targets for new interventions. We did stop that trial because there was a concern that the coronavirus might be transmitted through stool samples, and we couldn't rule out that stool donors were free of corona. So that actually stopped, but now there's much better interventions that you can do, which don't require taking 30 capsules. Um, there's been developments now where you remove most of the like excess fecal sample and you just focus on the microbiota. And so now it's like 2 capsules that you need for a transplant. There's also a number of other things that people are interested in, which is that, for example, if you were missing certain microbiota, could you develop probiotics that people would take? Or if there's metabolites that the microbes are making, maybe you just use the metabolites. There's some interesting work in Florida that's studying aging. And because we know that there's certain metabolites that are lost in aging, scientists are trying to replenish those for older adults in the form of like metabolite cocktails. There's also some interesting work to engineer bacteria. So perhaps you could engineer bacteria that produce a metabolite of interest or even a neurotransmitter. And you could see if you can design bacteria that would then have benefits for health. I have been told, though, that if you develop bacteria, that you also have to develop a kill switch because you don't want these engineered bacteria to take over and crowd out so-called normal uh, bacteria. Although, on the other hand, apparently it's also very hard to change the gut. So people who are engineering these are just in the first place trying to get them to work. And then, of course, diet and prebiotics, prebiotics being those things that feed the bacteria. These are all ways that we could modify the gut microbiome. There was a citizen science project called the American Gut Project that was active for a number of years. I don't think they collect samples anymore. This was led by Rob Knight and others. And in this particular project, what you could do is you could send in a stool sample and then you would get some information back from the project about your gut microbiome composition. And in return, the American Gut Project got a lot of stool samples across the United States, as well as self-reported health and dietary data. And what they found was that when thinking about diversity, which we generally think that gut microbiome diversity is the kind of healthy gut microbiome, the predictor was not things like, vegetarianism versus, paleo diet, or we could think of these like sort of drastic diet differences, but it was more about the number of unique plant species that people consume. And so the more plant species that you consume, and specifically people consumed 30 different kinds of plants per week, had the greatest microbial diversity. So if you want to do a little experiment for yourself, you could track what kinds of plants you're eating. And 30 is not necessarily that high of a number if you think about all the fruits and vegetables and different kinds of grains, beans, etc., legumes that you're eating, even herbs. Those might get you up to 30. Kind of coming back around to what we started to talk about at the beginning of this presentation, there was actually a clinical trial that wanted to see whether human immune status could be modified by modifying the gut microbiome. And what the investigators did was to test both a high fiber diet and also a high fermented food diet. And in the high fermented food diet, these were individuals who really weren't eating fermented foods, but then were encouraged to eat them. And that could include things like yogurt or kimchi or kombucha, other fermented food products. And the idea was to see if it would change the immune system, potentially in positive ways to contribute to health. Kind of going along with what Elia Mechnikov was interested in. And what the investigators found was that high fiber diet really didn't make much of a difference, which kind of goes back, away from many of the things we talked about here. But the high fermented food diet had a stronger impact on the gut. And it also had a beneficial impact on the immune system. And what was interesting was that in the individuals who had the high fermented food diet, it wasn't necessarily that they were getting more diversity of the gut or just more of those microbes that they were consuming. Like it wasn't just the microbes in the yogurt were now in their gut and were crowding out bad bacteria, like Metchnikoff thought. But rather that something about eating fermented foods actually remodels the gut. So it makes it look different and also more hospitable to microbiota, which in turn kind of allows your microbiome to be more diverse. So there is something about these fermented foods that are remodeling the gut that's quite interesting, and suggests that there are ways to change the gut for good health. So we have a number of, this brings me to the end, and I'm happy to take questions. I just wanted to share that we have some other things that we're working on in our program. We have now kind of stopped the fecal microbiota transplant project, but we've moved to doing a probiotic study where we have kind of a custom probiotic for Alzheimer's disease in partnership with International Flavors and Fragrances. We're going to continue seeing whether some of the genetic effects or genetic risk factors we see for dementia might act through the gut. We're studying Down syndrome because individuals with Down syndrome overproduce amyloid due to an extra copy of the amyloid precursor protein gene. And so we're testing whether they also have differences in gut microbiome. We're seeing whether family members transmit gut microbiota to each other that affect health outcomes. One of the big influencers of gut microbiome composition is actually geography and where you're living. And as I mentioned, I mostly talked about our human studies today, but many of the things that we're hoping to do are via mechanistic animal studies. So with that, these were the conclusions. I think we talked about all of these, but I wanted to acknowledge that there are a lot of people involved in this research, including our research participants who really provide so much information to us. And I'll take some questions. Thanks, everyone.
Thank you so much. This was an amazing presentation. It's also our highest attendance we've had all the way since we started the series for in July. So this was awesome. I'm gonna just leave the floor to people to ask questions. I'll start off with the one that was put in the chat though. So someone was wondering about if there are any differences by biological sex, whether that be in animal or human studies with the gut microbiome and how that affects dementia.
Yes, such a good question. And in the animal studies, the effects actually were largely seen in the male mice. And yeah, for sure, I'll show the last slide. In the animal studies, there's definitely sex differences, and in fact, some investigators were thinking that they would stop studying the female mice because they only saw the effects in male mice, which of course we don't want to necessarily do. But for some reason, with those germ-free models, the effects were seen in the male mice. In the human studies, we also see sex differences, and in some, I wouldn't say that there's any, pattern that's always the same. Sometimes we see that a metabolite is having more effect or relationship with the brain in women. Sometimes we see that another metabolite is having more effect on the brain in men. So there is definitely 100% sex differences that are very, very important. And for that reason, it's important to stratify by sex and look for sex interactions. There's also a student in our group who's trying to understand if there might be differences in women in menopause and with hormone therapy. So a lot of complexities to tease out, but definitely worth the effort.
And then I see a raised hand. Yeah, feel free to unmute and then we can go back to the chat.
Thank you, Barbara, so much for the presentation. It was wonderful. And we are very interested in, I work with Tom Perls at BU. We are very interested in making connection between gut microbiome, diet, metabolomics, and cognition. So we do a lot of work and we should talk more. But my question is, you mentioned that there are efforts to predict the diet of individuals based on the gut microbiome. And I wonder if it's known, what is the rate of remodeling of the gut microbiome? And if you predict the diet based on the microbiome, is this an idea of a average diet, or is the diet of the last week, for example, what
Oh,
people ate the last
yes.
week?
Yeah. And I will admit that my lab specifically doesn't do this work, but as I understand it, it is when they're looking at the metabolites, I think it is probably within the last week or last 3 days. So for example, if there's like metabolites that you see when you eat fish, then those would be, within the last few days. On the other hand, there also are a lot of metabolites that you can measure due to medications, and those persist much, much longer. I think even after someone maybe has stopped medication. So, so I think that answers your question, but I think for the diet, it is much more short term.
Mm.
We collect diet information via self-report using the food frequency
Mhm.
questionnaire. And, because we'd like to know, of course, the diet is so important. And what are people eating? What's their microbiome composition? And what kinds of metabolites are they then producing? Um, we use like a pen and paper type of collection because that seems to work best with our research participants, but probably using some more modern methods, using pictures of food and, stool analysis or ways to increase the rigor of that. I think probably
Mm.
that's where the field will go, but we do everything by pen and paper right now.
That's exact the same thing we have done in one of the last cohorts. So it's a lot of work, but and the data seems to be pretty reliable despite
Yeah.
yeah.
Yeah. And our participants, some of them don't have smartphones. And so we don't want to necessarily, exclude people who wouldn't be able to do the
Yeah.
assessment via computer. So
Yeah.
that's why we do it the way
Thank,
we do.
thank you so much. It's a very impressive work. Thank you so much.
Thanks. Nice to meet you.
Awesome. So there's a question in the chat. If there are any differences for those that were breastfed as infants versus formula.
Yeah, I love that question. And when we started our research, we created a questionnaire where we asked people all these questions that we thought could influence their gut microbiome, including were they breastfed? Were they born via cesarean? And we don't have enough diversity in our data to look to see whether, if you were breastfed as a child, would that influence your later life outcomes? Because that would be really interesting. But in the infant literature, there's definitely differences between the gut microbiome in breastfed and formula-fed babies. And apparently it's a huge industry in formula development to kind of optimize the formula for microbiota. So if anybody has ideas, then apparently that's a big business.
Got it. Yeah, awesome. And anyone else has any other questions, feel free to raise your hand, unmute, or put them in the chat. Um, while people are maybe thinking of their questions, I had one that I wanted to ask. So I know you were talking about the microbial metabolites, but I was wondering if you could speak a little bit to the role of the gut microbiome in producing and modulating neurotransmitters like serotonin or GABA and dopamine. And do you see that those effects are altered in Alzheimer's disease and individuals with dementia?
Yes. So that's not work that I do personally, but I know that there are other investigators in that are really interested in this. Um, there is this statistic, which I'm pretty sure is evidence-based, that's floating around that a lot of the serotonin, like that's produced in our body, is produced by the gut itself. We haven't measured neurotransmitters, but I think that, if you were working in animal models, that you could certainly measure those and they do play a role. So the answer is yes, but in Alzheimer's disease, we don't know if some of these effects are via the neurotransmitters, but a very good idea.
Awesome. Thank you. I appreciate that. If anyone, oh, there's another way. In the chat, any way to introduce fermented food bacteria into food that's not traditionally
Mm.
containing such?
Yeah, I think so. Because I think that there could be, you could introduce the the bacteria that you see in fermented foods and put them in other foods. I don't see why you couldn't. But I think the general, what I've heard from my colleagues in bacteriology is that you're supposed to eat the food itself. And I don't know if that's because it has the most bacteria or you wouldn't be able to achieve the same amount of bacteria, or maybe some of the bacteria in fermented foods are difficult to culture otherwise and put into other foods. I don't know. But it would be neat if you could enrich other foods, I guess. I like your idea, especially if you don't love kimchi or kombucha. But just to share with you, Federico told me that this is the tip. This is the takeaway tip of the day. He said, eat chia seeds because they're fiber rich and drink kefir, which some people also call kefir. But he said that those two things should keep your gut microbiome in good shape. So that's the health tip of the day.
Awesome. Love that. Perfect. Yeah, if anyone has any last questions, I will be sure to post this recording online for those of you who want to review it and send it to others. But this was a fantastic presentation. I just thank you so much for coming. And we look forward to seeing all the future work that you guys do at Wisconsin.
Thanks, Sammy. Nice to meet everybody. Take care. Bye-bye.
Awesome. Thank you.
Thank you.